Welcome to Trial Site News podcast series. We are excited to have June Round with us here today. She is a pathology associate professor at the Huntsman Cancer Institute, University of Utah. Today we will be discussing obesity and the immune system and how they might relate. So June, welcome.
Thank you, Adrian, for having me.
Yeah, it's our pleasure. We're excited to have you here, and everything you have to say, it's going to be fun. So before, though, we deep dive into our topic today, if you would, share with us what inspired you to become involved in your current research.
So my lab is primarily interested in how gut microbes that live in your intestines and on your body influence the immune system. Mm-hmm. So I got into this originally as a graduate student. I was trained as an immunologist, so really studying how the immune system functions. And at the time, this was maybe about 15, 16 years ago, we were really being trained that the immune system was supposed to get rid of bad microbes and that every time it saw a bacteria, it would just get rid of it. So I learned that a lot of these bacteria actually live on our intestines. And this made me really wonder, well, how does the immune system tolerate these organisms? Why does it let these organisms live on our body? So this is really what launched me into understanding how the microbiota influence the immune system. Mm.
Now, your research does reveal that there may be an immune system gene that could lead to or cause obesity and type 2 diabetes. Would that be an accurate statement?
So we did in fact knock out a particular gene that recognizes microbial particles, and we knocked it out specifically within a T lymphocyte. But the major consequence of knocking out that particular gene is that it disrupted the antibody response in the gut. So I wouldn't necessarily say that this gene's linked to obesity per se, rather that the way our immune system reacts against our gut microbes can lead to obesity.
Gotcha. Now, what was the negative results of that?
Of the gene knockout?
Yes.
As far as the immune system goes, the major consequence was that your antibody response in your gut was reduced. And then when you reduce those antibodies, you get an outgrowth of some of the organisms in your gut that are bad, which in turn caused the good organisms to be reduced. And it was that reduction in those good organisms that led to the obesity in our mouse model.
Oh, wow. Now, we are aware that you set up preclinical studies on mice to learn more about this phenomena at your University of Utah lab. You found that this genetic defect impairs some of the immune cells in mice and consequently leads to weight gain and some health problems. Could you elaborate?
So not only do these animals develop weight gain, they develop many of the same signs that we call metabolic syndrome in humans. So this includes insulin resistance. It includes, immune dysregulation of the adipose tissue. So you see a lot of inflammation happening within the adipose tissue itself. And you also develop signs of fatty liver disease in these mice. Now, what's more, even beyond the immune system defects, we found that all of these metabolic symptoms, they really stemmed from the type of microbial community that lived within the gut of these mice, such that if you deleted that microbial community, that you reduced weight gain, and if you added back those microbes that were lost, that you could also reduce weight gain.
Now, apparently shifts in the gut microbiome were linked to the immune problems. Is this true, and could you elaborate further?
Are you asking specifically in humans?
Both your mice and then humans, yes.
Yeah. So what's interesting about our mouse model is, so mice and humans have similar microbes in their gut. There's differences, but there's a lot of functional similarities between what microbes do in the gut of mice and humans. So what's interesting about our mouse model is that a lot of the differences that you see in the microbial community of our mice are recapitulated in people who have type 2 diabetes and obesity. So the biggest change in the microbial community of both mice and humans that are obese is that they have a decrease in the number of organisms they have. So that's referred to as diversity. So they have a decreased diversity of their microbial community. And what's more is they have a specific decrease in these organisms that are referred to as Clostridia. And that's seen in both our mouse model and humans.
Now, if what you have found to be the case in mice is applicable in humans, could it be possible to boost a person's immune system function, which might in turn prevent obesity and associated health issues?
Oh, that's such a good question. I think that it is possible. So this, as I mentioned before, this all came down to the antibody response in your gut. Mm-hmm. And these mice had a reduced antibody response. So it was kind of an inappropriate regulation of those microbes in the gut. Now, vaccination is something that we do very commonly, mm-hmm. To get rid of all of these bad microbes. And I think if we, of course, more research is going to need to be done, but there is a component of the microbiota that was, quote unquote, bad in our mouse model. There was an organism that really outgrew and caused the deletion of these good microbes. So I do think potentially it could be possible to maybe make an immune response against that microbe so that you can ensure that you control its growth in your gut and prevent it from outgrowing. So I do think that this could be a possible strategy to prevent the development of metabolic syndrome and obesity.
Oh, wow. Well, that would be interesting, because as someone who's fluctuated in weight over the years, never got obese or anything, but I went from being, like working out with football players, helping them train for the NFL Combine, benching 365 pounds to, not working out and then finding that, wow, gaining 50 pounds, and then you have additional, minor health issues that come up. And so that constant battle to stay, fit. Now I work at it, but it takes work. So this is fascinating for me. So let's turn now and let's focus on the genetic defect itself. What we know is that you uncovered the defect in the MYD88 gene. The immune system defect stops mice you found in the lab from making the MYD88 protein in their T cells. So as we know, T cells help the immune system recognize and kill bad bacterial and viruses. Can you elaborate on the MYD88 connection into your research?
Yes. So MYD88 is it's called an adapter molecule, but what it's really important for is signaling downstream of what are called toll-like receptors. And these receptors recognize pieces of bacteria or viruses. So essentially, these are receptors that allow the body to recognize something that is foreign, like bacteria. So the reason that we knocked out this particular gene is because all of these microbes that live in our gut also express a lot of these bacterial ligands that would stimulate these toll-like receptors and also stimulate MYD88. So MYD88 allowed us to look at, by knocking it out, allowed us to look at how this pathway might be responding to the microbes in the gut, and then what the consequences of that would be.
Ah, I see. Now, additionally, in regards to gut microbes, your research lab found that the mice you studied had reduced activity of some T cells called T helper cells. These helper cells tell the other immune cells, known as B cells, to produce microbe-fighting antibodies. But in the mice with a defective immune system, the B cells made less of these antibodies. Could you elaborate more on this and why?
Yeah, so that was a pretty good coverage of what we showed. The major defect is, so the T cell type that we saw was defective. It's called a T follicular helper cell. And these particular T cells, they hang out in lymph nodes, and they hang out in places in your gut called Peyer's patches. And as you mentioned, they help to stimulate those B cells and that antibody response. So we don't fully understand why, but those T cells are sensing microbial ligands, and this is helping them to help those B cells make antibody responses. We're currently trying to figure out, why that is. What are the signaling pathways downstream of MyD88 that activate this response? But we don't really understand that fully yet.
Now, your research lab revealed that altering the microbiome may impact how much fat is absorbed in the small intestine. Giving the mutant mice in the lab extra Clostridia reduced the production of protein involved in fat production. However, you also found that giving the mice disulfovibrio, I never get this one right. Disulfovibrio, okay. I can do that again. So, however, you also found that giving the mice disulfovibrio had the opposite effect. It increased the protein's production in the mice. We read that this, too, suggests that Clostridia protect against obesity, while disulfovibrio appears to promote obesity. Could you comment on this?
Yeah, so our studies, I think we did a fairly decent job of showing that those Clostridia are good at downregulating lipid absorption within the gut. What we showed in our study regarding disulfovibrio is that disulfovibrio itself is not causing the obesity per se. Rather, the outgrowth of this organism causes death of the Clostridia. So it's actually causing those good organisms to die or change their function. So they no longer put the brake on lipid absorption. So I don't know that disulfovibrio is directly causing obesity, other than it's doing it through reducing those beneficial Clostridia, if that made sense.
It does. So it's kind of
it is. It's like a microbe interaction that's causing it.
I guess it would be the secondhand smoke of obesity.
That's funny.
Yeah. See, this is what happens when I have my coffee in the morning.
Yeah. Clever.
So, we know that scientists are aware that both gut microbes and diet help to determine body weight. But the new study reveals that the immune system is vitally important in how this all plays out. Other researchers have raised the question about this research, such as why our microbiome is in charge of how much fat the gut absorbs. Could it be that increased fat absorption evolved to deal with having too much of a certain gut bacteria?
I guess I look at microbes as being inherently selfish. So for me, I don't know that we have any data to support this, but we had this idea that the reason that these particular organisms were decreasing lipid absorption is because they were taking those lipids and using them for themselves. And we have a little bit of data to support this, but we're continuing research along these lines. So if you were to ask me personally, why do I think this evolved? I think it evolved because the microbes, want some of our food for themselves. But as you point out, there could be a lot of other reasons why this happened, and maybe this is a great way to ensure that there is an appropriate balance of the right microbes in the gut, and that's something that we hope to figure out in the future.
Which would be amazing. Now, what is the path forward with your research? Do you see human studies in the short to intermediate future?
So the first thing we really have to figure out is what are these organisms? So when I refer to these Clostridia, it's really a group of organisms that represents about 50 different species. So when you think about a potential therapeutic use of these organisms, 50 is just too much. So we're currently working to whittle down, what are the critical organisms needed in order to protect from this metabolic syndrome. And right now we're at about 10 different organisms. So that's a little bit better for a potential therapy, is working with 10 different organisms. So we're continuing to work on that so that we can turn this into, a probiotic, if you will. Another area that we're also looking into is we did show that there's something secreted from these organisms that has this effect. So another potential therapeutic use would be to identify whatever that molecule is, and then we can make it perhaps, in the laboratory, so that could be a therapy in and of itself.
Gotcha.
So that's another potential area that we're looking into.
Okay, well, that's exciting stuff, and I know we'll be keeping an eye on it as it moves along. You're doing great work, and we're excited to see where this goes.
Well, thank you.
Yeah. Well, thank you so much for talking with us today, June. This has been enlightening, and I know we're excited here at Trial Site News to see how this continues to develop. And for me, on a personal level, always interested for better ways to handle weight.
Yeah.
So, and for those of you in the audience interested in learning more about this project, you can email June at june.round@path.utah.edu, and links will be provided in the description below. And you can also find us on Trial Site News on Instagram, Twitter, Facebook, and YouTube, and the links will be provided in the description below as well. So thank you for joining us today, June.
Thank you, Adrian.
Yeah.